very quickly. Okay, then we'll directly jump into the practical session. Okay, so what we are going to actually, this is 2 days workshop. Day one today. The session is, as you know, everyone is free to join. But tomorrow it'll be multi-ligand docking, which is a paid session. So if you want all the details, our WhatsApp group is link is shared in the first message. Okay, you go through that WhatsApp group, and we'll be sharing all the details. But anyway, so today we are going to go through basic molecular docking steps. Okay, from starting to the end, right? So it will cover the entire molecular docking process. So we'll do protein preparation, ligand preparation. So today we'll be doing single ligand docking. Okay, so we'll be doing protein preparation, ligand preparation, define the grid box, then run the docking, validate the docking protocol. If time permits, we'll visualize the uh, interaction, right? So we'll go through all this uh, complete molecular docking workflow. So you see, most of so we are trying to learn molecular docking. So it's perfectly normal if you are not able to understand the theory or the practical part for the first time. Okay. So if you have opted for the paid version, then we will be sending you with the PDF tutorial, the recorded video. Okay, so you can practice at your own pace. Or otherwise, if you are not opted, also it's not a problem. So every month we'll be repeating the same workshop. Okay, so you can join and try to learn again. Okay, it's not a problem. So if you are not able to understand the theory or the practical part, just this is just to show you the overview of how molecular docking works and also how you can execute the molecular docking steps. Okay, so we'll be just learning the basics. We are not going to go into advanced molecular docking workflow. Okay, so what you should not expect from this day one molecular docking workshop is that we are not going to go into the detail about uh, what is a search algorithm, what is scoring functions and all. Okay, these are the theory part, but it's important, but we are not going to cover this today. Okay, so again, flexible docking, metal ion docking, covalent and hydrated docking, this comes under advanced docking. So we are not going to cover this today. We are just going to do basic molecular docking, taking one single ligand and then do docking and see how the interaction as well as how you can validate the docking procedure. Okay, and then what are the required software? So when you install MGL tool, MGL tools, you will be also installing this Autodoc tools automatically. So bundle under MGL tools. So I repeat again. So we have also given you a video link in the WhatsApp group. There we have explained why MGL tool is one of the most important tool in molecular docking. Okay. So again, there are problems when you install MGL tools. We have also rectify or clarify like why it is not able to install in new systems. Okay, even though if it is not compatible with the new systems, still it is important because most of the paper in molecular docking are based on Autodoc Vina and Autodoc 4, right? So we have given the paper of the number of paper or number of publication using different software, and we have also explained in that video. Okay, so yeah, it's up to you if you want to use Autodoc tools, Autodoc Vina or Autodoc 4 in your docking research or in your molecular docking research. But yeah, this is free and you have to negotiate somehow to find and compatible system. Okay, so we have also defined, explained to you which are the compatible system for installing MGL tools and Autodoc tools, right? So yeah, so today's main engine or main software for running the docking is this Autodock 4. This will be getting as autodock4.exe file. So this is uh, command line kind of uh, or executable file. It doesn't have any GUI format. Okay, so yeah, so we are going to convert the but yeah, let's see how it goes. Okay, so if we have time, we'll just visualize and what I'm to do in for visualization, we'll be using Chimera X. Okay, so tomorrow for day two, this is a paid version. Okay, so this will be like day two paid one. So we are going to discuss about multi-ligand docking against a single receptor. So first, before docking, we will download a ligand library. We'll see how to create a ligand library. We'll download it from Zinc database. Okay. And then we'll see how to configure the input files. And then finally, we'll execute using Autodock Vina using command line for a single ligand first. We'll see how it processes. The same thing is done in Pyrex. Okay, but Pyrex gives you a very nice graphical user interface. You can do where you can do multiple ligand docking, but in the background, it is using Vina only. Okay, so before running Pyrex, we would like to show you how you can run Vina so that the background also you can understand, right? How Pyrex is running the multi-ligand docking. Anyway, so this we will do using Pyrex, and then we will be also generating a publication ready figures, 3D, 2D interaction images. So it will uh, just to show you the glimpse of. So we will be creating a figure something like this. Okay, this will be all free software we'll be using. In this figure, we use Maestro, but tomorrow we'll be using Discovery Studio, and similar to this format, we'll be generating a 2D interaction as well as 3D interaction. Here only 3D interaction is showing, but so we'll generate two interaction like magnified this thing. Okay, so one with 2D and one with 3D. Right. And again, see, for this also, we'll be providing you, this is again for the paid participants. Okay. So we'll providing you all the details about this molecular docking procedure. Okay. So if I go a little faster, or I mean, I will try to be very slow, but yeah, if it is a little faster to you, then you can always refer to the tutorial as well as the video. Okay. This is for day one. Again, for day two, we'll be doing ligand library generation. The same PDF tutorial will be given and for molecular docking using Autodock Vina command line as well as Pyrex also will be giving you a tutorial for that also. Okay, so yeah, I think this is good enough for the topics that we are going to cover, right? So let's come back to our topic again. So whoever is saying screen not visible, please exit the session and then join again. Just a second, okay? One second, I'm trying to find. All right. So yeah, something got stuck. That's why. Yeah. So you see, whenever we talk about drug discovery, Okay, so molecular docking, obviously it comes under the technique, like the particular topic, uh, drug discovery pipeline or something. Okay, so it comes under, sorry, yeah, drug discovery, right? Pipeline are saying that, uh, or we talk about drug discovery, we are ultimately trying to refer to a search for a therapeutic target, oh, therapeutic agent, right? Therapeutic, sorry for the spelling, okay? Therapeutic agent, right? Like drug molecule or so, potential drug molecules. Someone is saying, yeah. So those who are not able to hear my voice clearly, please exit and join again, or check if your net is stable or not. Everyone else is able to hear it clearly, okay? So we are trying to find a therapeutic agent or drug-like molecule or drug molecule that can help or treat, manage a disease condition, right? So before we dive into docking itself, let me take you back and let me ask a question. Okay, so what exactly is a disease? Okay, so I'm coming to this because I want everyone to understand the basic concepts before we jump into molecular docking, because in molecular docking we require this foundation. Okay, so yeah, so most of you, I think some participants are even teaching. So yeah, so please bear with me, those who already know, but many of you are beginners and not even from biology background. Yeah, so that's why we are just trying to give you a level playing field for everyone. So let's see what is a disease, right? So any condition that disrupts the body normal functions, which often are characterized by specific symptoms like fever, inflammation, fatigue, respiratory issues, etc. Right? So what causes a disease? There can be two factors. The one factor is this external factor, which involves infection through various pathogens such as virus, bacteria, and fungi. Right? So another can be through this internal factor. So they are primarily caused by body malfunctions, like including genetic mutation, which can lead to cancer, okay, autoimmune responses, hormonal imbalances, etc. Right? So what are the interventions? So disease interventions include medical treatments like taking medicines, and sometimes you have to go for surgeries, you do lifestyle changes, etc. Right? But most of the time when we are having a illness, or when you are getting sick, you take medicines in the form of a drug molecules, right? In the form of drugs. So let's see what is drug and how they work. Okay, so this is the here we are going to introduce some important concepts. If you are not familiar with this drug discovery process, okay, so please pay attention. Those who are not familiar with these concepts. So let's see what is the drug and how does it work. I'll just go through it and try to explain again. Okay. So a drug is a biological or chemical entity that can modulate the course of a disease state by interacting with a specific molecule, which is called the drug target. So by how they are going to interact and modulate the course of a disease state by either enhancing or inhibiting the function of this particular drug target. Okay, so which ultimately will alter the biological process and leads to production of some kind of therapeutic effect, leading to physiological responses that alleviates the symptoms or treat the disease condition itself. Okay, so let's see. So if you have a drug molecule here, so what is it saying? It drug can be like anything, like this two, mainly these two. They can be biologicals like vaccines, antibodies, etc. Okay, and they could be synthesized, right? Chemical entities. Nice. You can synthesize drug molecules, small molecules mostly, and there are also natural products. Okay, drugs from natural products. So what they do, they interact with this drug targets, right? So what are these drug targets? They can be enzymes, okay? They can be proteins, ion channels, etc. Okay. So when they interact, they can manipulate the function of this drug target by either what? Enhancing, okay? You can enhance the function of this drug target, or you can inhibit the function of this, inhibits the function of this drug target. So when you inhibit, this drug molecules will be called as antagonist. Okay, so these terms, if you read any paper related to drug discovery, you are going to find this frequently. Okay, so if if the drug molecule or potential drug molecule inhibits the function of this drug target, they will be called as the antagonist, or if they enhance the function of this drug target, they will be called as the agonist. They means this potential or anyway drug molecule. Okay, right? So when they interfere or manipulate the function of this drug target, this lead to some biological changes, and that led to therapeutic effect, right? So this can be in the form of alleviation of the symptoms, okay, of the disease condition, or complete treatment. Okay, so you have complete treatment of the disease, right? So everyone with me? Everyone? Yeah. So if you understand these concepts, then we go to a real example. Okay. So with that example, we'll see what is the drug target and why we want that drug target, and how we are going to use to, I mean, at least, manage the symptoms or treat the disease condition. Okay. Using drug molecule. How we can do that. Let's see. Okay. So let's take an example of Alzheimer disease. So what is this Alzheimer disease? Alzheimer disease is a progressive neurological disorder that causes the brain cells or neurons to degenerate and die. Okay, so what are the main symptoms? Memory loss, progressive impairment in rational thinking, and you can also have behavioral impairment. Okay, these are the main symptoms. Okay, what are the changes that you see in Alzheimer patients, okay, pathophysiology, right, inside the brain? So you see this neurofibrillary tangles. Okay, you see these neurofibrillary tangles, right? This is the normal. This is the Alzheimer patient or disease condition, right? So you also see neurotic plaque, right, beta amyloid plaque like this, and you can also see rapid loss of synapses and also basic cholinergic neuron. So these are the neurons, and synapses means the junction between these two neurons. Okay. So one more important observation that we find is in the Alzheimer condition or Alzheimer patient is that there are this neurotransmitter also. Okay, their level also get drastically reduced. Okay, this neurotransmitter level. So based on this, scientists came up with a theory called cholinergic theory. Okay, this was in 19 probably 1960s or 70s, something like that. Okay. So what this cholinergic theory says, these are the neurotransmitter, mainly this acetylcholine, okay, in the brain. So this is the neurotransmitter, and the neurotransmitter in the synaptic cleft, that is the junction between two neurons, their level get reduced in the case of Alzheimer patient. So this reduced condition of the neurotransmitter, because of this, you are observing those symptoms that we discussed or we have mentioned in the, sorry for the spellings, okay? So symptoms, right? This main symptoms. But now we know that it is not that because of this one parameters, or because of this one condition. There are many conditions, but at that time, so the theory was that because of this reduced condition of neurotransmitter in the synaptic cleft, you are able to observe those symptoms. Okay, so idea is, if you can maintain this neurotransmitter level in the synaptic cleft, maybe you may be able to reverse the symptoms. Okay, if because of this reduced condition you are observing those symptoms, if you can reverse the, or maintain the condition, that is that this neurotransmitter level, if you are able to maintain in the synaptic cleft, probably it might reduce, or reverse the symptoms, or you will be able to manage the symptom at least. So yeah, that is what, uh, I mean, let me go through one more time. So this is the acetylcholine, that is the neurotransmitter. So how they get transported, let's see its function first. So it is transported through the synaptic vesicle and released in the synaptic cleft, right? So once it is released in the synaptic cleft, it does its function, okay, transferred its information. Once the information is transferred, it is broken down into acetate and choline. These are the inactive forms, right? So forms of the inactive forms of the neurotransmitter. So by an enzyme called acetylcholinesterase. Okay, this is the short form of the enzyme. So this, okay, enzyme converts the neurotransmitter acetylcholine, which is in the active form, to inactive forms, okay, acetate and choline. So if we kind of block or inhibit the function of this enzyme, so if you are targeting this, this will be our drug target in this condition. So if you are going to target this enzyme, why we are going to target this enzyme? Because it is converting the acetylcholine into choline and acetate. So our main objective is to maintain the level of the acetylcholine. If you block this enzyme, obviously it's not going to break down this acetylcholine. Therefore, the level of the acetylcholine will be maintained in the synaptic cleft. Okay, the hope is that if you are able to maintain the uh, this level of the neurotransmitter or acetylcholine, maybe you may be able to reverse, or kind of manage the symptoms of the Alzheimer patient. So that is what exactly this donepezil does. This is an antagonist, okay, antagonist, okay, for this enzyme called acetylcholinesterase. Okay, so this drug molecule donepezil, it comes and interact with this enzyme acetylcholinesterase and inhibit the function of this enzyme. Once the function of this enzyme is inhibited, it it manipulates the biological function. I mean, it kind of, yeah, manipulate the biological process, right? So that means the neurotransmitter level is maintained in the synaptic cleft, and that is how it is able to manage the, or it is still used to manage the symptoms of the Alzheimer patient. So still now donepezil is used. Okay, so one of the popular drug molecule still now available in the market is donepezil to manage the symptoms of Alzheimer patient. Okay, so here is the,
Mm-hmm.
what is this? This is the experimental or crystal structure of this particular enzyme, acetylcholinesterase. Okay, and this is the pocket. Okay, this we will be calling this as active site pocket. Why we are calling this as active site pocket? Because the catalytic residues serine 203, histidine 447 reside in this particular pocket. Okay, so it forms kind of a channel. So this is donepezil, and it is how it comes and sit inside the pocket of this particular enzyme and block its function. Or otherwise, in the this natural condition, so acetylcholine will be the substrate. It comes and binds here. So once it comes and sits here, so this serine will become, nucleophile, and it will just do a nucleophilic attack. So once it does its function, this will, catalyze the substrate, and it will break down into choline and acetate. Okay, so if the drug molecules comes and binds here or interact with other residues, important residue here, now this acetylcholine won't be able to enter here at all because it will be blocked by this particular drug molecule. Okay, so some concepts are there. I mean, competitive innovation in the non-competitive and all. Let's not go into that. So it comes and binds here, and it will block the entry of your substrate. Okay, so that is how it is able to inhibit the function of this enzyme. And particularly this pocket, will be calling this as the active site pocket, because the active residues which, or the active site residue is here. Okay, so it resides in this pocket, and this is very important because it does the catalytic function. So if you understand this concept, let me go to molecular docking. And using this concept, let's try to explain what is molecular docking and how we can apply molecular docking to find alternative drugs, okay, or a possible drug molecule for this enzyme. Okay, so let's see what is molecular docking. So molecular docking is a technique to predict the ideal orientation, affinity, and interaction of a ligand in the binding site of a protein or enzyme, right? So what it does, one, that is orientation. It kind of predict the orientation of the molecule that you are going to, see if they can fit inside the pocket of your target enzyme, okay, or protein, right? So once you have this ideal orientation, you will be able to calculate the score, some kind of predicted binding affinity or some kind of free energy binding, okay, free energy of binding, something, okay, that can give you a score, right? You will have a score. And you can also visualize the interaction between the ligand and the residue at the binding site or the active site pocket, something like this. Okay, so this is the surrounding residues, and this is your molecule, and you can see what kind of interaction it is having. This is like pi-pi interaction, cation-pi interaction, some hydrogen bonding interaction, etc. Okay, so depending on what kind of interaction it has, we can extract very important information which will be useful for our studies. Okay, so what kind of studies? Let's see. Okay, right. So the same target drug target we have here, the zoom in version here, okay, and this is the pocket as we have mentioned before, right? And this is the drug molecule. So just to understand what is docking, let's assume one thing. Let's assume for this, I mean, demonstration sake, okay, just to make you understand, okay, let's say that this drug molecule donepezil is giving you a serious site activation, effect, or in short, it gives you a very bad toxicity. After using it for decades, we come to know. I'm not saying that is, actually showing you this side effect, okay? But let's assume for now that donepezil is giving you a very side effects. So taking donepezil instead of managing the symptoms, if it is giving you a very serious side effect, it's better to not take donepezil, right, or the drug molecule. So what will be the next step, or what will be the next study, okay, next research? So scientists obviously will be looking for replacement of this drug molecule. How do we replace this drug molecule, or how do we find drug molecule which can show similar kind of binding affinity, or binding interaction, or some kind of fitting inside this pocket showing similar kind of effect as the original drug molecule. Okay, that will be the case, right, with less toxicity. So in that case, you can, if you have in your lab some 100 molecules or so previously synthesized, okay, you can take those molecules and do some experimental in vivo or some kind of in vitro studies, right? So you can